I always loved the ocean since I was a kid. I grew up in Baltimore, Maryland, which is right on the Chesapeake Bay. And so I was always really interested in the ocean and marine life. But I do remember trying to watch as many marine life documentaries, seeing one about dolphins and whales where there was a scientist explaining something. And the title under that scientist was biological oceanographer. And since about 10, that was what I told family and friends I wanted to be when I grow up. I did not know exactly what a biological oceanographer was. I did not know how it was different from a marine biologist, but that is what I told people my career aspirations were. I'm Dr. Colleen Petrik, and I'm an assistant professor at Scripps Institution of Oceanography at University of California, San Diego. My research is 100% what we call in silico. So that means it's all on the computer. And so I use computers to build little simulation oceans, little marine ecosystems where I'm representing all the animals and the physics and the different processes that link them together. And the computer iterates that forward so I can see how things change over time. And by doing it on the computer, it means that I can manipulate different parts of the ocean that would be basically impossible to go out and do in the real world. When I'm not sciencing, I'm spending time outside because I sit in front of a computer all day for research. I like to go to the beach and just relax and read on the beach. I could sit there all day. I like to take my dog to Dog Beach or hiking. And I like to travel. My husband and I travel a lot in search of craft beer. We also drink a good amount of craft beer here in San Diego. It's one of the cities known for craft beer. I can't even begin to fathom what a world would look like without science, because science is how we understand things. So everybody would be walking around doing something without knowing what the repercussions are, or guessing what the repercussions are, or maybe not even caring. And you know, for coastal systems, we humans have a large impact on the coast with land development, with agriculture and nutrient runoffs in rivers, as well as toxins in rivers, coastal development, building, all of that. And I think if we didn't have science, we would drastically shape the coastal marine ecosystems negatively and probably lose a lot of organisms and a lot of unique habitats and food webs. I'm Dr. Colleen Petrik. Welcome to the Jeffrey B. Graham Perspectives on Ocean Science lecture.
Thank you so much and welcome. Today we're going to be talking about science and seafood, so let's dive right in. And most of the seafood I'll be discussing tonight are fish. So how many fish do we catch worldwide? I'm going to show you a chart, and the Y units of this chart are in millions of tons. And that's a unit of mass or weight that we don't commonly use day to day. And it's also really big numbers, and we people are not great at big numbers. So put this in perspective, 1 ton, T-O-N-N-E, is equal to 1 metric ton, T-O-N, very confusing, equal to 1,000 kilograms. Gotta love the consistency of that metric system. But basically, that is the weight of the amount of fish that could fit in one of these boxes that you see on the deck of a fishing boat here in Alaska. So they fill these boxes up with fish and then take them to be processed. So that's 1 ton. But our Y units are millions of tons. How big is that? Well, it's roughly the amount of fish that would fit into one football stadium, like the University of Miami Orange Bowl shown here. So, starting at 1950, which is roughly our modern industrial era, we were catching 20 Orange Bowls worth of fish. And you can see that this fish catch, these are wild capture fisheries, has grown over time. And from 1950s to the 1980s, it has increased. And then starting about the 1990s, it's really stabilized somewhere between 80 to 90 million tons. However, if we look at human population, that continues to increase over time. And we would think if there's more people, then there would be more fishing and more fish catch. but maybe this suggests we have hit the upper limit on how many fish we can catch from the ocean. And so this might be problematic. One reason this mismatch is problematic is that people rely on fish as a source of protein. This map is indicating with color the amount of a population's dietary protein that comes from fish. So the areas shown in orange have a really high reliance on fish for protein. And as populations increase, but fish catch does not, it means we might not be able to meet the food security needs of all these countries. Similarly, people rely on fish for economics. Here in the US, our commercial and recreational fisheries bring in over $300 billion and support over 1.7 million jobs. Does anybody know what the biggest US fishery is, or the fish that we catch the most of here in this country? These are good guesses, but it's pollock. Pollock, you say, Colleen, I have never seen that on the menu at my seafood restaurant. It is not at the fish taco stand down the street. What is pollock? You have eaten it. Yes, ma'am. It is fish sticks. It is a McFish sandwich. It is imitation crab meat. And if you think about the prevalence of these products, then you understand how this is our biggest fishery. So let's learn a little bit about pollock. As their name suggests, they live in Alaskan waters, specifically the Gulf of Alaska and the Eastern Bering Sea. They look like this. They look very similar to a cod. If you've ever seen a cod, they're in the same family, and they support our very large fishery. I studied the population of pollock in the Eastern Bering Sea. And when I was studying Eastern Bering Sea pollock, I was really interested in the early life history stages, and specifically this transition from being larvae and juvenile to becoming adults. So these new fish becoming adults are important because they influence the size of the adult population, which is the fish that we catch. So here I'm going to show you how the numbers of new fish entering the pollock population have changed over time relative to the mean. So above this 0 line is above average new fish recruiting to the population, below average. And you'll notice it's highly variable. There are a lot of high highs and low lows, and very rarely is it near that mean value. But there doesn't seem to be a pattern over time, just the fact that it goes up and down. So one might ask, like I tend to ask, is what is causing this variability? Could it be related to the environment? So let's look at one measure of the environment, the temperature, here in the pink line, also shown relative to its mean. We're seeing it goes up and down too, but maybe there are some changes over time. It seems to have increasing magnitude in its range, where the highs and lows get bigger, and it also might be increasing over time. So this is a different pattern from the pollock new fish entering the population. So maybe these are not really related. We could try squinting at this and see if any patterns arise. And actually, if we start concentrating on the late 90s into the 2000s, I start to see some relationships. So in years with above average new recruits to the population, that seems to happen when temperatures are cooler than average. Whereas years with below average new recruits to the population happen when it's warmer. And we can see this pattern much more clearly if we just plot these two variables against each other. And our numbers of new fish decrease as temperature increases. That's a negative relationship, a negative correlation. So this is usually where my research starts. Somebody else will find this pattern, and then I step in to answer why and how does temperature affect the number of new fish entering the pollock population. So to answer these questions, I really have to find the mechanisms underpinning this relationship. And to do that, I use mechanistic models. So let's take a step back and discuss what is a model. Unfortunately, we're not talking runway models tonight, but we're talking about models that you are intimately familiar with and probably use in your daily life. It is just a method for describing how the world works, and there are many types of models. One is a conceptual model, a formal description of some aspects of the world for understanding and communication. A good example of this from oceanography is how thermohaline circulation works. We call this the ocean conveyor belt. So this is the movement of water masses around different parts of the global ocean, driven by density, which is made up of temperature and salinity, hence the word thermohaline. So this shows you how in some parts of the ocean we have water moving, say, northwards in the Atlantic from the equator up to Greenland at the surface, and then it cools and goes down, and it's now that deep blue color, and that's the bottom water moving south towards Antarctica. This is an oversimplified representation of how water moves around the ocean basins, but it's not wrong, and it's a good starting point if you want to learn the more specific details or just have a basic understanding. Another type of model is a physical model. This is simply a smaller or larger physical copy of an object, sometimes to scale, other times not so much, like our solar system models that we made in elementary school. And then finally, we get to another type, mathematical models. And these might be a little scary because they are based on equations. We use equations to describe a system or a process. So Einstein used this equation, E equals mc squared, to describe how energy was related to mass times the speed of light squared. Whereas Newton's second law of motion says that force equals mass times acceleration. A type of mathematical model is a statistical model. This is based on empirical observations that represent the data generating process of the system. What does that mean? Well, we could say that this relationship that is tying new fish and temperature together is linear. And that means that the data generating process is going to create observations that fall along a line. And the way that we would test that with a statistical model is to fit a line to these observations. And the method of doing that is linear regression. So if I fit a line to these data, I get a line with an intercept and a slope. And here we can see this slope is negatively related to temperature. Another type of mathematical model are mechanistic models that I use. These have many names like numerical, simulation, or computational models. And this is where we imitate a real world process or a system and how that operates over time. This is how we generate weather forecasts or hurricane models. These are numerical models. So we are taking all our understanding of atmospheric and oceanographic physics, like laws of thermodynamics and Newton's laws of motion, and stringing them together and then iterating them forward in time to see the evolution of, say, the movement of atmospheric air cells to create winds that create a hurricane. So now that we understand what models are, why do we use them? What are they useful for? Well, particularly for oceanography, there are a couple great things about them. One is the ocean is ginormous. We can't measure everywhere at every time. If I want to go out and measure dissolved iron, this is a micronutrient that's really important for phytoplankton. Or if I want to go measure zooplankton biomass, these are predators on phytoplankton, then I need to get on one of these big research vessels. I need to go around to different sites that are of interest to me, throw things over the side of this boat, well, lower them very carefully because they're expensive, and collect water in bottles or collect plankton in nets. Do this in sites and at times that are interesting. Then bring those samples back to the lab and measure the concentrations of the iron, put the plankton under a microscope and identify them to species level and then count those. And it's a lot of work. It's a lot of time, effort, and money. And what we get then are measurements of dissolved iron around the globe and zooplankton biomass around the globe, but it leaves this patchy, sparse picture of the whole ocean. Maybe you can see some patterns here. If we look at the zooplankton biomass, the high biomass in red seems to be appearing in the North Pacific, and maybe it gets lower into those blue colors near the equator. So we think maybe there's a latitudinal pattern in zooplankton biomass. And we see that in the northern hemisphere, but does that pattern exist in the southern hemisphere? There aren't really enough data here to see. Similarly, with iron, it looks like there are high concentrations in the tropical equatorial Atlantic, but does the Pacific also have high values? So this is where models come in. They can fill these observational gaps. And now we can see the whole pattern and how these two quantities vary from region to region. And now we can see that, no, these patterns are not the same. Zooplankton biomass is different in the northern hemisphere versus the southern hemisphere, and iron is different in the Atlantic versus the Pacific. Another reason to use models is the ocean is complicated. There are many interconnected processes happening all together, and it's hard to disentangle their individual effects and to quantify those individual effects. For example, if I have a very simple food web here made up of two food chains, one in the upper waters and one in the seafloor, and I want to understand what is the influence of primary productivity, the base of the food chain, on my fish populations. Maybe I'd like to perform an experiment where I increase the amount of phytoplankton and the productivity and see what the effect is. And so we will get the downstream effects of everything related to phytoplankton, which will be increasing zooplankton productivity and increasing benthic invertebrate productivity. These processes are interconnected to the phytoplankton. But maybe that's not really my true question, and I want to know the individual effect of increasing zooplankton production or increasing benthic production. With a model, I can actually do these manipulative experiments to see what the result is and quantify it. And then finally, another great reason to use models is the problem with time. Time is always moving forward and at the same rate. We can't observe the future and we can't observe the past. But we can use models to forecast or predict future conditions. We can also use models to learn where things came from. So here I've got a little schematic showing a hatch area for fish eggs and where those eggs go in the ocean as they develop into juvenile fish. That would be running our model forward in time to forecast or predict. We can also run it back in time if we collected some juveniles and we want to know where they were spawned as eggs. So we've got models that do that as well. All right, so we know what is a model and why they are useful. How do we actually go about modeling marine ecosystems? Because I already told you, the ocean is complicated. This is just one food web in one region, and there's so many different organisms up there that we can't even read them. And there are lots of lines connecting them. So I don't model the ocean ecosystems like this. This is too complicated. Let's make things as simple as possible so that they are still useful, though. And let's think about the animals that live in the ocean. How would we represent them? They do the same things that we do. They eat, they breathe, they grow, they reproduce, and they die. So if I want to know how many fish are in the ocean or the amount of fish in a population and how that changes over time, then I just need to focus on the amount going into the population and the amount lost from the population. And we can break that down. What are the gains? It all comes from eating. But there are different sources of loss. There's breathing and other waste products. There's reproduction and death, getting eaten by a bigger fish. So here, we're just back to a conceptual model. saying that the change in biomass of a fish population is equal to what they eat minus the losses from respiration, reproduction, and mortality. But now, how do I change this conceptual model into a mathematical model, a mechanistic model? Well, I have to have equations to represent these words. And how do I do that? Well, let's focus on one example here, the eating aspect. How do fish, or really most predators, feed? The way we understand that is from lab experiments. So you might think that the feeding rate of an animal would be related to different things about its environment, like the temperature, or how much prey there is, or what type of prey there is. And then it might be related to the fish itself, like its size. So we perform these experiments in laboratory settings where, for instance, we put a fish in a tank with some prey. And this would be to study how ingestion rate, or the prey eaten per hour, varies based on the number of prey. And so you can see from these little schematics that we collect some data, and generally it looks like it's increasing. If we put them all together, yeah, we do see that increasing pattern. That the more prey in the tank, the greater number of prey that are eaten per hour. So still, this is a pattern, but this is not a mathematical equation I can use in my model. To get that equation, we have to fit curves to these data. So you could make different assumptions about what type of curve to use. One would just be a straight line. As prey increase, so does ingestion rate. But a lot of studies have shown that we actually taper off our ingestion rates, and not just fish, but really any predator. Zooplankton, land animals, people. That there's really two components. There's searching for your prey, which increases as the prey increases, but then you also have to handle it. So fish aren't using hands to catch their prey and pull it in their mouths and chew on it, but they still have a handling time. They have to orient themselves to the prey. They have to suck it in and do whatever processing of swallowing, etc. And then, orient themselves to the next prey. And so there's just a limit to their ingestion rate, no matter at the highest prey densities. So there's lots of support for this equation. That's the one I'm going to use in my model. I simply plug it in here. And then I start doing that with every other aspect, having an equation for respiration, for reproduction, for mortality. And that gives me how I model this population of fish. And then I can go about modeling other populations of fish that are slightly different. So in orange here, I've got a small pelagic forage fish, like sardines and anchovies. Those live in the surface waters and eat plankton. I also model pelagic fish predators, these large-bodied predators like tunas and billfishes that also live in the upper waters, but they eat prey on fish. And then we have demersal fish predators that live near the sea floor. And they eat things like benthic invertebrates and also fish. This would be our pollock in the eastern Bering Sea. So as I mentioned, we've got a governing equation for how fish biomass changes. It's related to the amount of prey in the environment. It's also related to temperature, so I need temperature conditions, and it's related to the size of the fish, and that I have baked in. I have assigned sizes to them. So I get my temperature conditions and the amount of plankton prey from another ocean plankton model, and then I run my fish model using those results. If I use the oceanographic conditions from the recent historic time period, let's say the 2000s, this is what the model estimates the global biomass of fish are. So here, these maps, again, I'm showing blue representing low, the kind of cyan and greens are intermediate values, and red is high. So this is the amount of biomass of each of these types of fish, and you see they have different patterns. Our small forage fishes up in the top left are pretty widespread, but their predators, these large pelagic fish, have a much more restricted habitat. They are high in productive regions and absent from very cold regions, but also absent from very hot regions. And then finally, our demersal fish at the bottom are really high abundance in cooler areas like temperate subpolar regions in the high latitudes, and also high biomasses near the continents in shallow shelf environments. So this is a global picture that is just showing us the differences of our food webs. But if we really want to understand the food webs, we can just pick individual locations and look at the structure of those food webs. So I've got our little fish pictures again in our food web, but it's really hard to scale those to accurately represent the ecosystem. But instead I have the actual quantitative values coming out of the model in these bubble plots, where the size of those bubbles are scaled to the biomass of each of the organisms in that food web. And the width of the lines connecting them is the flow of biomass from one animal to another in that food web. And so you can see that these food web structures are quite different. Here I've got 3 examples from the Pacific. Starting on the left, we have the Eastern Bering Sea. We should be intimately familiar with this place now. It's cold, it's shallow, and it turns out that's really good conditions for demersal fish, that big green bubble, like our pollock. If we go to the middle and we look at the coast of Peru, it is a upwelling system, very much like our California current ecosystem here. So it's deep, and you have deep, cold, nutrient-rich water coming to the surface, supporting a lot of phytoplankton and zooplankton productivity, and thus we get a lot of fish that live in those upper waters too. You can see the orange bubble is a good size and a really big blue bubble, so a lot of pelagic fish. And then finally, in contrast, we can go to the middle of the Pacific where the Hawaiian Islands are. These are really warm waters that are low in nutrients. And what we see are just fewer biomass altogether of the plankton and the fishes. So this is setting the stage. This is creating a framework for my understanding of how the environment structures marine ecosystems. With this understanding, I can then perform some experiments. So I'm going to focus on looking at that time aspect of the utility of models and forecast the future. So a lot of my research is to do climate change projections. To do this, we have to make assumptions of how the climate is going to change. And we know our climate is changing because of the concentration of greenhouse gases in the atmosphere. So people create these scenarios, they're showing up in different colored lines, of the amount of greenhouse gas emissions over time. And so our red line is kind of a worst case scenario that is assuming we have the same emission rate as today, but per capita. So as the number of people increase over time, those emissions are really going to grow. And you can contrast that with the blue line, where we take a lot of climate mitigation practices, say we convert to all renewable energy and have very low emissions going into the future. And then there are 2 scenarios in between. This is the effect of those scenarios on sea surface temperature around the whole ocean on average compared to what it was in 1900. So that's why it starts at 0 and goes up to as high as 5 degrees Celsius if we look at those uncertainty bands. We can look at other aspects that come out of these models. They're not just simulating temperature, they can simulate the phytoplankton and their productivity under these different greenhouse gas emissions scenarios. Here again, you can see these different colored lines, red, green, yellow, and blue, though they don't have quite as clear of a separation, and these envelopes of uncertainty are overlapping a lot. It's not a very clear picture. But I'm just going to focus on one of these scenarios, which is more or less the worst case or the biggest, most drastic changes. And that's where the temperature has gone up really high, over 3 degrees Celsius, and the phytoplankton productivity has dropped down low, close to 10% decrease. And not surprisingly, when we use this emissions scenario with our fish model, we see that the fish biomass also decreases in response to the changes in temperature and productivity at the base of the food chain. However, you will notice that the amount of decrease is not the same across these three types of fish. Our green demersal fishes, the ones that live near the seafloor, have the smallest amount of change. They're a bit buffered from the climate effects because they are at the seafloor where the water stays cold, away from the surface warming. Whereas our blue, large pelagic fish have the biggest change. They live in the upper waters where they're hit by that warming, and then they're also a top predator, so they're influenced by the decrease in production. So this increased warming is increasing their metabolic demands. They are hungrier. But there's less energy at the base of the food web that is getting to them as a top predator. So they suffer the most. So this is a long-term projection into the future, but we can also do shorter-term projections. We call these forecasts. And yes, I do guarantee 100% humidity tomorrow. So you are familiar with forecasts. You use them if you look at the weather. Maybe you never look at the weather. It's San Diego. It's basically this every day. But our weather forecasts start with observations. We say, what is the temperature, the wind speed, the humidity right now? And we use those as the starting conditions for our weather model. And then we let the weather model run freely without any information from the observed system and just do its thing based on the physics equations. And then that's going to tell us the temperature on Wednesday or on Saturday. We can do the same thing with our oceans. We can create forecasts of the temperature or the plankton or even the fish biomass. So here I'm going to show an example of a forecast of total fish biomass, specifically in this region off the US West Coast, the California current ecosystem. And you might be looking at the date on this plot. It says 2013, and think, Colleen, I thought you said forecast, and that's supposed to be the future. Well, the problem with forecasting the future with a model is that you can't really judge if it's any good because you have nothing to compare it against. So we have to forecast the past. We call these retrospective forecasts, and then we can compare them to observations and see how our models did. So here I've got a retrospective forecast that started in November of 2011. So I've got a map showing the distribution of biomass of all those fishes combined relative to the mean. Purple values are above average, orange values below. And then on the right side is a time series of that biomass in this forecast, also relative to the average. So in 2012, we're starting out above average. It goes for a couple of years, staying around that average with some seasonal variation. And then I'm stopping here in 2015 because something anomalous happened. Does anyone remember anything different about 2015? A huge marine heat wave, I heard. The blob is what we called it. We had extremely warm temperatures, much warmer than average in the North Pacific. This little map is showing the temperature above average, where yellow is a little bit higher, and the redder you get, much warmer than average. And so you can see some really red values along the west coast of the US. This resulted in changes in the ecosystem. There were declines in krill and forage fish populations. There were harmful algal blooms that created shellfish poisoning, and marine fish species moved hundreds of miles northwards. So let's see how the forecast did in 2015. Look at that, we see a big decrease in biomass, much like we observed. So it is capturing some properties of the system. It was able to forecast this decline in fish biomass. So that's the change in time. What about the spatial aspects? So here's now our distribution of fish. Maybe this doesn't look any different from the last one, so let's put them side by side. Now hopefully you can see some of the changes. We've got a lot less purple, especially in the southern part of the map. And so that is that contraction of the biomass to the north. So we are also matching those observations where we saw a northward shift in fish. So this looks really good. It shows some promise for being able to forecast fish biomass. And so then what could we do with this fish biomass forecast? Maybe we could use it for making fisheries management decisions. Right now, fisheries management does forecast fish biomass, but they do it with statistical models. And they're usually just based on properties of that population. They're not even incorporating how the environment changes. Except for one, the Pacific sardine stock here off of California does get harvest regulations set in relationship to the environment, specifically temperature. So the exploitation rate, or the harvest rate, the fishing rate, is related to the March temperature anomaly. So here we see a curve that as that temperature anomaly increases, the exploitation rate increases too. This is because these warm waters have traditionally been better for sardine. So the warm waters, we expect a bigger sardine population so we can catch more of them. And this rule is based off of the past 3 years' temperature. And the reason that we use the past 3 years is we are assuming that next year is going to be the same as the past 3 years. But what if instead of assuming what next year's temperature is going to be, we can forecast next year's temperature instead? So we used an ocean model to forecast that temperature. Here I'm showing it now with a different color scheme. Purples mean low values, yellows mean high. And then again, we do some retrospective forecasts to see if there's any skill in our model. We can assess this skill by comparing it to observations with a correlation coefficient. What you need to know is that the redder, the better, the more skill. So this is the skill of a forecast in temperature 1.5 months into the future. It's very skillful. If we go even further into the future, 6.5 months, you can see the reds have kind of dulled. They're a bit more medium in color, but there's still skill in some regions. So we have potentially skillful forecasts of temperature. Let's use that to set our sardine harvest. Well, before we go ahead and make any changes to how we harvest sardines, we have to show that this is better than what we were doing before. So a colleague of mine, Desiree Tomasi, and collaborators, Desiree works up the street at NOAA, went on doing this test. Is incorporating a forecast of temperature better than the status quo? So as a control, they used Harvest Guideline 1, HD1, just constant fishing effort, no use of temperature. Then compared that to the current Harvest Guideline 2, using the observed temperature over the past 3 years to estimate the fish biomass and set harvest rates. The third scenario, using only 2 years of observed temperature and adding a forecasted year, estimating the fish biomass and using that to set harvest. And then finally, a slightly more sophisticated one, where they're using 2 years of past observed temperature, 1 year of predicted temperature. But now instead of just estimating this year's fish biomass, we're going to estimate this year's and next year. So why might that be helpful? Well, let's say this year's fish population is going to be really high. That means we're going to fish a lot of them. But then next year's population is estimated to be really low. By fishing a lot this year, we're leaving not that many fish, and then they're going to get hit by some bad conditions next year. So instead, we might want to be more conservative this year, leave more fish than we typically would, so that they can help sustain the population under the bad conditions next year. How do these different harvest guidelines do? We can assess them in 2 metrics. The size of the fish population, the biomass, we can think of this as being sustainable. And then we can also assess the fishing yield, What is the economic benefit of using these rules? And ideally, we want to maximize both these things. And that's what happens with Harvest Guideline 4. It has higher biomass than Harvest Guidelines 1, 2, 3, and a much greater fishing yield. So it looks like we have skillful forecasts of the marine environment, and they have the utility to improve our fisheries management. And now this is a new direction that NOAA Fisheries is undertaking with the Changing Ecosystems and Fisheries Initiative. So I will summarize here. I use mechanistic models to understand marine ecosystems. These mechanistic models are an imitation of how a real-world process or system operates over time. Models allow me to fill in observational gaps, test processes individually, and forecast or predict future conditions. And with models, I have learned how climate affects marine ecosystems, how it sets the stage and determines food web structure, as well as how biomass changes under climate change or extreme events. And ultimately, the goal of my research in using models is to create information that can help make decisions in fisheries management. So with that, I would like to acknowledge my funding sources, especially NOAA. A lot of my funding comes from the Modeling, Analysis, Predictions, and Projections program, which is under the Climate Programs Office. They have been really amazing at supporting this type of research about how climate variability and climate change influences marine ecosystems. And NOAA is fantastic. They do more than fisheries. They do weather, they do hurricane forecasts, they do thinking about sea level rise. And so I just really want to acknowledge them and hope that you recognize their importance. And if you do recognize your importance, you can tell your Congress people that they should keep funding them the maximum amount possible. Thank you.
Do you utilize the Scripps collections at all in your work?
Do I use the Scripps collections? Yes. Well, I don't really go into the collections myself, but a lot of those collections come from observations made by the CalCOFI program and the LTER in the California Current Ecosystem, and those observations are critical to the modeling work we do. So I showed how modeling is useful for filling observational gaps, but we really need the observations for basis of comparison to see how well our models do. And then also these models, they are mathematical representations. They have these things called parameters, and some of them are really hard to pin down, and we can get more information about those from observations.
In addition to oceanography, one of my other interests is in medical research, spinal cord and neurological. And talking to the researchers in those fields, they've said a lot of the advances in the last few years have been greatly aided by advances in imaging and computing power. Have either of those things affected your work?
Absolutely. The computing power, for sure. I have showed you maps that may look really coarse in their resolution. We are increasing the resolution of our ocean models exponentially because of computational increases. And that means we can represent some smaller scale features of the ocean that are really important for biology. It also means we can run our simulations for longer time periods, or we can just run more simulations like they do for weather models or especially hurricane models. You've probably seen those spaghetti plots. If you run lots and lots of different simulations, you get a better understanding, a better prediction of the path of that hurricane. And so that computational power allows us to create that spaghetti plot, but now for our ocean ecosystems.
Yeah, this is all very fascinating. Let's talk about pollock and cod. North Atlantic, the cod population has really crashed, whether that's due to ocean temperature, environment changes, or overfishing. You can talk about that a little bit. And are we looking at maybe the same kind of thing happening to the Alaska pollock population?
So that is a difficult question because I'm not sure where the research stands right now with Atlantic cod. They were certainly very heavily overfished. Then a moratorium was put on the fishing, so we don't know why they haven't rebounded. That suggests something environmental. However, the pollock have always been very sustainably fished and have not been overfished. And I do actually have a couple slides to tell you what's happening there. So here's a plot of temperature at the seafloor in the Bering Sea in warm years versus cold years. So these pink, purple, and blue colors are what we call the cold pool. And these are waters below 2 degrees Celsius. The pollock adults avoid these temperatures. So when they are spawning, they are not going to go into the cold pool. So in cold years, they have very limited spawning regions off to the side along the continental shelf slope break there, whereas in warm years, they can really go all over the eastern Bering Sea to spawn. And that results in their eggs experiencing different currents. In the warm years, when they're on shelf, they get currents that are moving onto the shelf, and in cold years, they get currents that are going up to the north. And so this results in the eggs and the larvae and the juvenile fish going different places. These are now plots of observations of where these different stages of pollock were found in warm red and cold blue years. So this is another way to look at the distribution. Instead of the center of the distribution, it's the whole distribution. At the top, we have juvenile pollock abundance. Yellow is low, red is high. And this might not be what you expected because I said warm years were bad and cold years were good, but you can see the abundances are different. We then want to look at what they are eating, the zooplankton, and that abundance in warm versus cold years. I don't see a huge difference when I look at just the abundance, but instead, what if we plot the energy density of this zooplankton? And that's, whether we have a salad or a cheeseburger. So if we look at this now, we can see all the cheeseburgers are down on the southwestern area extending up to the north, which is exactly where the pollock are. But in warm years, we basically have only salad, and that's bad for the pollock. They're not able to accumulate enough lipid stores over winter. And so we can see that with this simple schematic from Sidden et al. In warm years, there's a mismatch in where the energy-rich prey are and the pollock are, but in cold years, they're overlapping. And that is what has been setting the stage for pollock in the eastern Bering Sea.
This is a very interesting presentation. I had a question and wondered how you see advances in AI impacting your work.
Yeah, I also think AI is another tool that's going to be great for filling gaps. So I have showed you some examples of climate simulations, and there were 4 scenarios. But what if we want to know something different from those scenarios? What if we want to have rapid rise that then gets a drop in 50 years in the future, a curve rather than an increase? And that's going to take a lot of computational power, a lot of hours and days of running those simulations that maybe we won't be able to get all the information we want on all the multiple scenarios. Instead, we can train AI models on these numerical models and then use them to predict the scenarios in between.
In your chart that showed that as the temperature increases, the biomass of the larger predators decreases more quickly than the pollock that are swimming deep. Would the fact that those predators are warm-blooded have anything to do with the way they interact with the temperature increase?
Well, I'm actually modeling everything as cold-blooded here, even though we think of tunas as being a little bit of both. Most of our fish are what we call ectotherms, cold-blooded animals, so that as temperature increases, so do all their rates, which is different from warm-blooded things like marine mammals. So the fact that they are cold-blooded means that they have this huge impact of the warming temperature, whereas something like a marine mammal might not be as influenced as strongly.
I know you've been speaking about Pollock, but I'd like to kind of change the species a little bit because we're talking about sustainability. I read recently a report, I can't remember exactly where it was, but there was a salmon farm and a storm came through and a great number of those salmon on the farm were able to escape into the wild. And that's going to have a major impact on the salmon population at some point because it's not the first time that's happened. I just want to get a comment from you about that.
Yeah, so I'm not particularly knowledgeable about salmon farms. I do know that aquaculture has its problems. Salmon seem to have a problem with salmon lice, so you got to worry about that spreading to the marine population. But in all these aquaculture, or even some of the kind of different aquaculture, not the whole life cycle, but the salmon in the Pacific, we take there as juveniles and then release them into the adult population. You still have to worry about those effects of the mixing of kind of your aquaculture ones versus wild, because your aquaculture really is selecting much more narrowly for specific genes and genotypes and phenotypes. So you're having less diversity in that population compared to the wild population. And diversity in genes is really good. It allows adaptation and evolution. And so if it happens that the aquaculture species become the dominant fish in that population, and then we lose genetic diversity, that could be really bad, that the population might not be able to adapt to novel conditions.
Thank you all for coming.